everyone, my name is Mita and my friend Esther. Today we are here to tell you about permanent thesis. Amita will start with introduction of permanent thesis. Permanent thesis is a thesis that is the result differentiate primary and secondary meristem cells. Permanent thesis is non-meristematic, does not grow close, not develop. Next, so what is the thesis? Thesis are group of cells that work together to do a job in the body. The group of cells that have the same form and function. Different theses can work together for a physiological function that makes up an organ. In the plane, the thesis is divided into two groups, meristematic and non-meristematic. Meristematic are theses in planes in the form of collection of stem cells that are active in cell division. This network is easier to find at the growing points of stem and root. Meristematic are divided into three types, namely apical meristem, intercalary meristem, and lateral meristem. Non-meristematic theses, namely theses whose cells do not divide anymore. Permanent thesis is divided into two, simple and complex. Simple consists of one cell type, parenchyma, collenchyma, and sclerenchyma. Complex has more than one cell type, or xylem and phloem. We will discuss about simple thesis. The first, parenchyma. Parenchyma thesis or basic thesis is a thesis consisting of living cell with varying morphological and physiological structure and still carrying out all the activities of physiological process. Parenchyma cell, a denwood cell that form the structure in many non-woody plants, including stem, root, and leaves. It is known to form the parenchyma of the cortex, or main filler, or the stem, the leaf. Parenchyma network has several characters. The first character is originating from protodrome and root meristem. Can be seen in the picture. Protodrome is the thesis that will become the epidermis. In the differentiation era, organogenesis process has run perfectly so that the epidermal layers has been cleared, differentiated, and has root here. Root help play a role in absorbing minerals from the soil because the differentiation process first occurs in the era. The differentiation era is called primary network. Root will cause elongation of root cells and stem growing point. Its characteristic feature is that it has denwoods because the cells are still young, facilitate the division process. Totipotent is the ability of cells to reproduce themselves. In the pictures above, there are various types of cells, semicolor, oval, and polygonal. The next characteristic can be seen in the image of elongated or labelled cell of various branches. In the polygon as the metric image, the cell walls are evenly coated on all sides of the walls. it can be seen in the figure. The thickening of the cell walls is only at the corners of the space between cell and not evenly across the surface of the cell walls. There are also some cells that define intercellular space that are almost closed because the canning of the lattice parenchyma network fill the spaces between the tissues. Parenchyma tissues is present out part of the plant body such as stem, root, and leaves. Parenchyma tissues leaf is located in the leaf mesophyll, normally palisade and sponge. Both tissues contain of chloroplast. This is a picture of a decayed leaf. There are non-palisade parenchyma and spongy parenchyma. In the palisade parenchyma like a post fencing, in the palisade parenchyma there are chlorophyll cells. Green leaf matter. The fencing parenchyma serve as a photosynthetic seed. The spongy parenchyma on the leaf serve temporary storage area for the result of photosynthesis in the form of gas, namely oxygen. Also applicably to suck carbon dioxide of it be used the photosynthesis process. Be found in pit mesophyll of leaf, cortex of stem, and roots, flowers, fruit, seed, xylem, and phloem. The image is visible cross section of the pedal nerve spot. The parenchyma is in charge of storage in this large pocket, a portion of cross cell with large cavities that help maintain excess water in the plant's aquatic habitat. Parenchyma with large intercellular space and branch is known as a parenchyma. The difference between parenchyma and sclerenchyma cell in the collenchyma chloroplast containing in the collenchyma chloroplast may be present or absent within the cells. Its diametric cell is a trine in the parenchyma that takes many forms. In transverse section, the cell look elongated and angular. Sclerenchyma cell network inert by the root. The cells are inert, soft, uniform, dense cell wall, hard, uniform, layered cell wall, photosynthetic and stronger in function. Photosynthetic and mechanical is function. Function strong storage of food, root and stem, cortex, pit, and pericyclum and phloem permanently to support quietly plants to feed on water. As parenchyma, photosynthetic color, palisade, and sponge. Parenchyma can be active to divide cambium and help the plant in secondary growth, formation of cord, and healthing of wood. Right, let's move on to new topic with Esther.
Next, with me, Esther Veronica Situmel. I want to tell you about collenchyma and sclerenchyma. First, collenchyma. Collenchyma comes from Greek, referred colla, that's meaning thick glistening cell wall. The origin of collenchyma is developed from the ground meristem or from procambium, example Apium graveolens. Collenchyme is simple tissue, that's meaning single cell type. The shape of collenchyme are range from round, elliptical to elongated cell. The collenchyme arrangement are more or less compactly arranged as the thickening materials deposit more heavily at the corners and on the radial walls. Collenchyme has similar characteristics to parenchyme, but thickened cell wall. The thickening is unevenly distributed. Cell wall composition are cellulose, hemicellulose, and pectin, but absent of lignin in lignified cell wall. Occupy the peripheral layers of cortex in the coats and beneath the epidermis or below a few peripheral layer of parenchyme. First, angular collenchyme. The thickening materials deposit at the angles or corners of the cell. In cross-sectional view, the thickenings occur at those places where several cells meet. You can find in the petiole of cucurbita and stem of S. tuberosum. 2. Lamellar or plate collenchyme. The thickening at tangential cell wall, which are found in cortex of stem of Sambucus nigra and petiole of Colellaria armoracea. 3. Lacunar or tubular collenchyme. The thickening materials deposit at cell wall which are in direct contact with intercellular spaces. Example, petioles of Salvia, Malvia, Asteraceae, etc. From the figure, the narrow shows intercellular space. 4. Annular collenchyme, which is defined by Duquesne, as it possesses circular cell lumen in transversal section. From the figure, the narrow show a circular cell lumen. And next, this figure show the location where the collenchyme exists in various planes. The collenchyme can be found around the cell as well as on the edges of the cell.
Next, the function of collenchyme. The function of collenchyme are: first, serves as mechanical support to the organ. 2. Chloroplast existence involved in photosynthesis. 3. Manufacturing sugar and storing it as starch. 4. In some cases, the peripheral thick walled collenchyme becomes thin and regains the meristematic activity. Example, telogen, the core cambium, which divides to form the periderm.
Right, let's move on to sclerenchyme. Specific features. Sclerenchyme usually that cell, sclerenchyme. Heavily thickened secondary walls and lignified, contain lignin. Rigid cell or unstretchable. Sclerenchyme arrangement compactly have an intercellular spaces. The main shapes as fibers and sclerates. First, sclerenchymatous fibers. The fibers is developed from meristematic cell. The fiber type are long, spindle-shaped shells with more or less thick secondary walls, and they usually occur in strains of lignification various. Fiber is parenchyma tissue comprising of fiber cell. The fiber are present in hypodermis of monocot stem, in pericycle of many dicots, in secondary root, and hypodermis of many leaves. Fibers is economically precious that are divided into hard and soft fibers. The pictures show an overview of the fiber. And then overview primary phloem fibers from stem. And next, and this is the overview of xylem fibers. At the last, this figure of phloem fiber. Two stone cell or sclerate. They are lignified, extremely thick walled. They are generally found in hard parts of the plant. Example, endocarp of walnut and coconut. The shape can be isodiametric, irregular form, and ramified. The cell can be single in plant tissue or in group in certain tissue of plants. In most plants, sclerate is idioblast, that's distinct cell possessing distinct size, structure, and wall thickness among the other cells. In Magnolia, the cell is found in most organs. This figure shows sclerate stone cell and branch sclerate. First, brassicosclerate or stone cell. That shape isodiametric and located in phloem, cortex bark, and mesocarp of Pyrus communis. 2. Macrosclerate or stick cell, which is elongated and cylindrical. It's found in the inner layer of seed of Leguminosae. 3. Osteosclerate or bone cell, that elongated with large ends like humerus bone. The sclerate is found in hypodermal layer of some species of Leguminosae, fruit and leaf of Ceropeae. 4. Asterosclerate or star cell. The shape structure like star and can be found in stem of Ceropeae and petiole and mesopyle of Camellia sinensis. 5. And the end, trichosclerate or filiform or columnar or polymorphic. That's long branch sclerate with pointed and spurred in intercellular spaces. It's found in hydrophytes that are floating organ of Eichhornia crassips, roots of Nipaceae, and petioles of Pistia. Okay guys, that's bring me to the end of our presentation. Thank you for your attention. It was pleasure being here.
Sorry if there is a wrong word or attitude. And finally I say bye-bye.